Hi, my name is Nikolai Van Oris, and today I'd like to give you a little story about the brief history of vaccines and how they've really benefited humankind. So the story starts with smallpox. Smallpox is a virus caused by a double-strand DNA, and it infects individuals, ultimately leading to very high fevers and skin infections. And the skin infections are pretty pronounced. They start as these little small red lesions that then start to rise as bumps, become fluid-filled, and this becomes a very fluid-filled blister that forms, scabs over, and then eventually sloughs off, leaving a large amount of scarring. Well, the first historical evidence of smallpox actually was seen in an analysis of 3 Egyptian mummies who were found to have a small smallpox-like rash, and this occurred in the 3rd century before the Common Era. So the earliest written description of smallpox was in the 4th century in China, and in the 10th century was really when vaccinations first took place. These weren't known as vaccinations, and what occurred in this case is individuals who had been exposed to smallpox and developed these scabs or lesions, the scabs were picked, ground up in a fine powder, and reintroduced in an uninfected individual in an aerosol fashion. And this ground up extract was able to then protect these individuals against disease. Now, this was pretty dramatic because there was a 30% death rate in those infected with smallpox. And in the 20th century, it's estimated that 500 million people were actually killed by the smallpox exposure. So this prevention was a unique strategy to prevent smallpox infections. Now leap forward to a period of 1750 to the 1820s. There was a physician named Edward Jenner, and he made a really important observation. What he noticed is that herdsmen and the milkmaids who would milk the cows would come down with smallpox-like lesions, but these were much smaller and not as severe, and these were called cowpox lesions. But interestingly, those individuals were never susceptible to smallpox infections, suggesting that that initial exposure with cowpox limited the effects of a smallpox virus. He took that information and then, using the sorts of techniques that the Chinese and the Arabs were started using, he extracted pus from one of these smallpox lesions from a young girl named Sarah Nelms, who was a milkmaid, and he took that material, ground it up, and then inoculated in this picture. You can see a small boy named James Phipps with this extract. Then, what can't be done today but was done then, is he infected this boy in 3 separate occasions with smallpox, and the boy never developed a disease. This then led to the introduction of these extracts from cowpox to prepare people so that they wouldn't succumb to a subsequent smallpox infection. So how do we know this actually works? Well, it's pretty obvious in the following picture. These are 2 boys. They actually look quite related. One was unvaccinated. This is the child on the left. And one was vaccinated. Both were exposed to smallpox, and you can see the dramatic difference. The child on the left doesn't look very happy. He has all these scabs that have formed throughout his entire body, and these will eventually encrust and flake off, leaving a lot of scarring. The boy on the right, totally protected from smallpox. So this is an example of vaccination, where one has taken an attenuated or a changed form of a cowpox, which is related to smallpox, used that to introduce it to an uninfected individual, and that individual is actually protected from the subsequent smallpox breakouts that occurred. And again, I remind you that for those who were infected and not vaccinated, the death rate was about 30% of those. The person I wanted to talk to you about is Louis Pasteur. So he was born a year before the passing of Edward Jenner, and he, of course, is best known for having discovered a process called pasteurization, where we heat different, in the time, milk and milk products to kill any of the bacteria that were in those to protect individuals. He also coined the term vaccination following those phenomenal results by Edward Jenner. So Louis Pasteur was also very interested in developing different types of vaccines in order to protect individuals. And one of the vaccines that he developed was something against rabies virus. So rabies virus is caused by a viral infection and usually involves a rabid animal such as a dog biting an individual. The individual gets the virus, which travels to the brain, and in the brain, of course, it initiates an immune response. So much like the flu, an individual who's bitten by a rabid dog will have fever, discomfort, very uncomfortable, and over time they'll become very agitated, sometimes confused. They've reported to have hallucinations, and they also have a fear of water. So what Louis Pasteur did was isolate this virus from a dog, and he introduced it, and you can see this in the picture, into the brains of anesthetized rabbits. And he passaged this virus over 20 times, which means he took it from one rabbit to the next to the next. And what occurred over time was the virus would change a little bit, or it would adapt to survival in the rabbit. So having this virulent or dangerous virus now growing in the rabbits, he would then take the spinal cord, which contained virally infected cells, and he would hang this up over a period of time, and the spinal cord would dry out, and in effect, the virus was completely inactivated or neutralized during this drying period. So he took that dried material, introduced it into rabbits in different doses, and none of them came down with an infection. But when he now took the virulent form of the virus and infected those rabbits, they were protected. So this was a second example by a different scientist of introducing a vaccine or the process of vaccination to protect an animal against disease. So this continued on, and at one point, there was one day a young boy that was brought to the Pasteur Institute named Joseph Meister. He had been bitten many times by a rabid dog, and Louis Pasteur agreed to try this dried spinal extract injection into the boy to see if the boy could be protected before he came down with rabies symptoms. So the way Louis Pasteur did this is over a period of 2 weeks, he injected the boy with extracts from those dried spinal columns over 12 consecutive times. The last time the boy was injected, he was injected with the virulent form of the virus, and he never came down with the disease, and he in fact never came down from any rabies symptoms. So this was the first development of a rabies vaccine that was successful in a human. So Louis Pasteur, after having told others of this success, went on to inoculate 350 other people who had been bitten by rabid animals. And in all cases, this dried extract that he had developed was actually suitable to protect them from disease that would occur with rabies virus. Jump forward to another debilitating disease caused by poliovirus. So poliovirus is a single-strand RNA virus. First enters through the intestinal tract, and then it spreads through the nervous system, and it causes inflammation of the spinal cord. And this leads to something called poliomyelitis, which is a disease which paralyzes the muscles. And this was such a debilitating disease for those who were infected, it led to the development of an organization called the March of Dimes. And their goal was to identify, treat, and then prevent this paralytic disease from occurring in young children. And in this little caption you see here, this is a young child where he had to use these braces to support his leg functions because the muscles had been so badly damaged. Other children who were infected with poliovirus had such damage to their lung muscles that they couldn't breathe on their own, and this led to the invention of the iron lung, which was basically a bellows that would expand and contract, providing them with oxygen from the air. So 2 scientists in the United States developed 2 distinct vaccines. One was Jonas Salk, And what he did is inactivate the virus, so it was completely neutralized and could never ever grow. And he used this to inoculate children who hadn't been exposed to polio and showed it protected them. Second scientist, Albert Sabin, actually developed an attenuated form of the virus. Attenuated means it can grow a little bit, but it's not very infectious. And he developed it in the form of oral droplets, which could be easily administered to young children. And there's still famous pictures showing these lineup of children with their tongues out, sticking out, waiting for a drop of this oral poliovirus vaccine. And this also protected them. The next person I wanted to tell you about was Maurice Hilleman. He was a researcher and he developed over 46 vaccines. He had an incredible knack for being able to work with a virus and identify inactive or attenuated strains of viruses that would be effective as a vaccine. He also developed a combination vaccine called measles, mumps, rubella, MMR, which is given to every child today. And this one was designed as a combination so the children wouldn't have to receive 3 separate vaccines, but could receive a combination. Again, this was a very effective means of preparing an individual to mount an immune response against either of these 3 viruses. 6 of the vaccines that he developed are still in use today in children during their childhood immunization schedules, and it's estimated that the vaccines that he has developed save on the order of 8 million children from dying each year. And these 6 vaccines, along with 4 others that form the toddler and child vaccine schedules, protect children from over 14 different diseases. There's a fantastic video that was developed about Hilleman. It was termed A Perilous Quest to Save the World's Children, and it provides a beautiful explanation of how these vaccines saved so many people. But the big question is, what is happening to the immune system with these immunizations? And the key to this question is that the immune system has memory and specificity. So what do we mean by that? Well, a vaccination or an immunization, and as I described earlier, an inoculation, all meaning the same thing, is the process where an individual is exposed to an attenuated or inactivated virus. It can also be a bacteria that's not infectious. And this comes from a virus or a bacteria, if it is normal, would cause a severe infectious disease. So the immune system detects this introduced material as a foreign invader, and it mounts an immune response. And that immune response is effective at killing any of the remaining virus, destroying it, and eliminating it. And this is fantastic and easy because the virus, in the form that it's given, is already neutralized and damaged, inactive or attenuated, so the immune system has a great opportunity to kill it. The key is that the immune system actually remembers that virus or that bacteria. And the concept is, when the real virus infects an individual, if that person has already seen that virus as an attenuated or an inactive strain, the immune system is ready. And what I mean by ready is that it rapidly mobilizes its functions to kill virally infected cells or to neutralize the ability of these viruses to penetrate into cells. The best analogy to this is thinking about a military campaign where you have information that another country is going to invade you. What you do is you mobilize your military, you provide them with armaments, you train them, and you determine what is likely going to be the enemy group. So, when the enemy does invade, your immune system is already mobilized, trained, and ready to respond, so it can effectively neutralize and prevent the virus or the enemy from creating a serious problem. Today, of course, we're dealing with coronavirus, which is a massive pandemic that struck the entire world very rapidly. And for the immune system, what one needs is a vaccine that provides the immune system with an effective means of killing this virus. And it can do that in two ways. One, we have this virus which has these very sticky spikes on its surface, shown in red. And if we can introduce an inactivated version of this virus, the immune system will make antibodies that will bind to these surface receptors, and the antibodies will effectively neutralize the ability of this virus using these spikes to enter into cells to infect them. The immune system also generates a population of cells called T cells, and should this virus enter inside a cell, the T cell is designed to kill components of the virus that are re-exposed on the surface of our own infected cell. So it might take a year or so to develop an effective vaccine, but the key is to develop an attenuated or an inactive virus that still has the same sort of surface features and characteristics that the normal virus has, so that the immune system, when it initially responds to the vaccine, will remember these different features. And then when the real virus comes along to infect us, we are ready, mobilize the army, and kill off the virus. Well, thank you for listening to this series, and hopefully we'll create some more interesting seminars on the immune system.